Welcome back, guys. Now let's discuss about how exactly this insulin is released. Okay, please look at here, guys. Here I am showing you the beta cell of the pancreas. So this cell which I am showing you right now, see, this is the beta cell. Now this beta cell of pancreas, see, it have prepared the insulin molecule. Okay, the insulin molecule is already prepared. Now this insulin, this hormone is stored in these granules. Okay, these are the granules in which the insulin molecules are stored. So these insulin containing vesicles are present in the beta cells of pancreas. Now my question to you, when do you need this insulin in your blood? When this insulin should be released into the blood? Whenever blood glucose levels are elevated, then you need to release the insulin, so that insulin will decrease the blood glucose levels. Okay, so now let's start our story from here. Now you have taken food. Okay, you have taken a good meal. Okay, now this food is going to be digested. Okay, now after digestion, you will be having glucose molecule. So after digestion, you will be having elevated glucose in your blood. Okay, there is postprandial hyperglycemia is there. Okay, now glucose is there in the blood, but where do you need this glucose? This glucose is supposed to be present in the tissues. The glucose was supposed to, the glucose should go into the tissues, but not in the blood. Okay, we don't want the glucose in the blood. We want the glucose to go into the tissue, so that tissues will utilize the glucose for the production of ATP. Okay, now see here, guys. Now we have the glucose in our blood. So glucose is there, hyperglycemia is there. Now insulin. Now this insulin should have to be released. Now the insulin should have to be released. So how this insulin? Okay, how these insulin molecules are released? That's the question. First of all, how this beta cell? Okay, how this pancreatic beta cell? How it knows that there is hyperglycemia in the blood, more glucose in the blood? How it knows? How it knows is, see, the glucose will enter into the beta cell. See, the glucose, this elevated glucose is there, right? This glucose molecules. Now these glucose molecules, they will enter into the pancreatic beta cells. Okay, so whenever there is hyperglycemia in the blood, whenever the blood glucose level is more, now this glucose will enter into the beta cell with the help of a transporter called as GLUT2. Okay, glucose transporter type 2. So this is an example of facilitated diffusion. Via facilitated diffusion, glucose is moving from high concentration. High concentration means, yes, in the blood there is more glucose present. So glucose is moving from high concentration to low concentration. In the beta cell, less glucose is there. So glucose is moving from high concentration to low concentration. But here this carrier molecule is helping. This carrier protein is helping. So who is this carrier protein? Nothing but GLUT2. So what is the first step? The first step in insulin release is the movement of glucose from the blood into the beta cell via GLUT2 transporter. Okay, which is an example of facilitated diffusion. Okay, sir, well and good. But before this, let me tell you something about the beta cell. Beta cell is inactivated, I should say. Okay, beta cell is actually hyperpolarized. It is sleeping. Okay, it is sleeping. So why I am saying this is sleeping? Because see, all the time this beta cell of the pancreas, see, all the time it is throwing potassium ions. These are the potassium ions. So the potassium ions are going out of the cell. See, potassium ions are going out of the cell. Okay, all the time it is happening. So potassium is going out of the cell. Why? Because potassium concentration is more inside the cell. We all know that the cells are the bags of potassium. So potassium is moving from high concentration to low concentration to so all the time potassium is going out of the cell. So if positive charges are going out of the cell, means cell is hyperpolarized. The cell is inactive. Okay, cell is inactive. Now what happens? See, now, yeah, you take the food, it's digested. Now glucose molecules are produced. Now the glucose came into the blood. Now this glucose will enter into the beta cell with the help of GLUT2 transporter. Now what happens to this glucose? Now glucose is going to enter into the pathway of ATP synthesis. Now first glucose will undergo glycolysis. After glycolysis, there is oxidative decarboxylation of pyruvic acid, followed by a Krebs cycle, followed by electron transport chain. So this glucose molecule is entering into the pathway of glycolysis and subsequent biochemical reactions. Okay. Now this glucose at the end of the day, okay, after electron transport chain, it helps in the production of ATP. So, okay, now in this beta cell, ATP is produced. Now what these ATP molecules are doing? Now these ATP molecules, they will go and they will inhibit, see, they will go and they will inhibit the potassium channels. Okay, so whenever the ATP levels increases inside the cell, the potassium channels are closed. So these potassium channels are called as ATP sensitive potassium channels. Okay, these are ATP sensitive potassium channels. Why we are calling them as ATP sensitive potassium channels? Why? Because these channels are closed whenever the intracellular ATP levels increases. So ATP sensitive. Now what happens? Potassium starts to accumulate inside the cell. Okay, potassium is accumulating inside the cell. See, potassium is a positive ion. So when the positive ions are accumulating inside the cell, what will happen? Cell will undergo depolarization. Cell will undergo a depolarization. Due to accumulation of potassium, there is depolarization. Okay, now this depolarization, whenever the cell is depolarized, what happens now? The calcium channels, okay, when the cell is depolarized, the voltage gated calcium channels, they are opened. Okay, the voltage gated calcium channels are open. So now what happens? See, the calcium starts to enter into the cell. Calcium is coming into the cell. This is the third step. Okay, first step is glucose entry. Second step is closing of the ATP sensitive potassium channels. Once the ATP sensitive potassium channels are closed, means cell is undergoing depolarization. Once the cell is depolarized, what happens? Now calcium starts to enter into the cell. We all know calcium is very important for the vesicular transport. Okay, here the vesicles are there. But once the calcium comes into the cell, calcium will go and bind with the secretory granules, and the secretory granules are moved. Okay, now the secretory granules, they will come and fuse with the membrane. Okay, now they will come and fuse with the membrane. Now what happens? Now the insulin, whatever is present over here, now fusing will happen something like this. Okay, the vesicle is fused and the insulin molecules are released into the blood. Insulin. Now see, now insulin came into the blood. So this is how insulin is released. These are the steps in insulin released. Now let's see one by one step. Already we have discussed. Just to make it more clear, see, 4 steps are there. Step 1, step 2, step 3 and step 4. What is the first step? It's the glucose entry. Whenever you eat food, food is digested, glucose levels enters into the glucose levels increase in the blood, and that glucose is going to enter into the beta cell of pancreas via GLUT2 transporters. So glucose enters the pancreatic beta cells through GLUT2, which is an example of facilitated diffusion. And now what this glucose is doing? Glucose enters into the pathway of glycolysis, Krebs cycle as well as electron transport chain. So glucose, at the end of the day, it generates the ATP. Now what this ATP is doing? Now this ATP is step 2, closure of ATP sensitive potassium channels. So ATP inhibits the ATP sensitive potassium channels, leading to reduced efflux of potassium. Means less potassium is going out, which means more potassium is accumulating inside the cell. When more potassium accumulates inside the cell, what happens? Cell will undergo depolarization. Okay, cell will undergo depolarization. But one point which I want you to know at this level, okay, one point, later we will discuss more detail about it. See, there are drugs called as sulfonyl ureas. Okay, glyburide, glipizide, glyclazide, these are the drugs called as sulfonyl ureas. Now these sulfonyl ureas are the drugs which are used in the treatment of diabetes mellitus. What they will do? Now these drugs like sulfonyl ureas, they will come and block the ATP sensitive potassium channel. Now see what the sulfonyl ureas are doing? Sulfonyl ureas are blocking ATP sensitive potassium channels. Now what happens if the ATP sensitive potassium channels are blocked? Means if the ATP sensitive potassium channels are blocked, means potassium is starting to accumulate inside the cell. Now this potassium accumulation will cause depolarization. Depolarization will lead to influx of calcium. The influx of calcium will cause exocytosis of the vesicles containing insulin. So insulin is released. So more insulin is getting released. So can I call the sulfonyl urea drugs as insulin secretagogues? Yes. If you take sulfonyl urea, more and more insulin will be secreted. So more insulin will be there in the blood, which is used in the treatment of diabetes mellitus. So for the treatment of diabetes mellitus, you can use drugs like sulfonyl ureas, which will block ATP sensitive potassium channels, leading to more secretion of the insulin. Next, see, let's come back. Now what is happening? Yeah, potassium channels are blocked. Because of that, potassium starts to accumulate. See, what is the third step? Opening of the voltage gated calcium channels. Why voltage gated calcium channels are opening? Why? Because see, accumulation of potassium ions leads to depolarization. Okay, so depolarization opens the voltage gated calcium channels. Calcium ions enters into the beta cell. Calcium ions are entering into the beta cell. Okay, now what this calcium will do? See, the fourth step is exocytosis of the insulin. So entry of the calcium ions causes the exocytosis of the vesicles containing insulin. Okay, so these are the four important steps which are involved in the exocytosis or in the release of the insulin. Hope the video is helpful. Thank you.